In this video we shall understand the genesis of resting membrane potential. As you already know that all excitable tissues have a potential across their membranes, meaning if we insert one microelectrode inside of the cell and the other outside and connect them to a voltmeter, then a negative potential is recorded by the instrument. This potential is called as the resting membrane potential. The negative potential means that the inner side, or intracellular fluid side, of the membrane is relatively negative compared to the outer side, or extracellular fluid side. We need to understand the genesis of this negativity of the membrane potential in the resting state. We all know that the intracellular and extracellular fluids contain molecules in charged state. These are known as anions and cations. The chief cation in ICF is potassium ion, whilst in ECF is sodium ion. Whereas the major anion in ICF is protein, which is abundantly present inside the cell and has lots of negative charges. Chloride ion is the major anion in ECF. When the cell membrane is in resting condition, that is in absence of electrical impulse to cell, another process known as Gibbs-Donnan equilibrium operates in the cell. According to this equilibrium, whenever a semi-permeable membrane separates two chambers, here ICF and ECF, containing a mixture of permeable and impermeable anions and cations, then, the side containing impermeable anions will end up having a slight excess of positively charged ions. In case of cells, intracellular fluid contain abundant proteins and they are not diffusible through the cell membrane, so, ultimately ICF will end up having a slight excess of cations, which will be potassium ion. There are several leak channels present on the cell membrane in the resting state for diffusion of both sodium and potassium ions. Normally potassium leak channels outnumber sodium leak channels in a ratio of 10:1. It is then, this excess potassium ions that try to diffuse from ICF to ECF down the concentration gradient in resting state. To counter this flow of positive charges outside the cell, sodium ions try to move from ECF to ICF again along its concentration gradient. But this flow of sodium ions is limited because of lesser number of sodium channels present in the cell membrane. Because a slightly more number of positive ions has diffused out compared to positive ions diffusing inwards, the negative charge present on proteins inside the cell gets exposed. It is this negative charge which manifests as the negative resting membrane potential.